As we take a look at Chapter 9, one of the things you'll notice is there's an awful lot of information here. This is certainly one of the biggest chapters in the book, and so we're going to break this down into 3 different audio presentations today, starting off with kind of an introduction to genetics and looking at Mendel. Then we'll move on to some ideas beyond Mendel, and then the last part is looking at some specific human examples in terms of disease inheritance. So as we begin to move into our genetics area of our semester, a good place to start is with defining a gene. And a gene is just a basic unit of inheritance. We think of it as kind of a chunk of information that's going to tell our cells how to build proteins. And so certainly genes are composed of DNA, and Chapter 10, our next chapter, will look at that chemical structure of DNA and how it works. But the idea behind a gene is that it's going to provide information on either how to build a particular protein or when to build protein. Remember, we've got over 250,000 different proteins as humans that we have to make. Each protein has its own sequence of amino acids that has to be just right for that protein to fold in its correct shape and have its correct function. And so genes are going to basically provide that instruction booklet on how to build a particular protein. And if we look at a particular gene in one of our cells, we all don't have the same genes. And so those different versions of a gene are called alleles. And this is a really important term we'll use an awful lot here in Chapter 9, this idea of an allele, or different versions of the same gene. And so, for example, something like tongue curling. Whether or not you can curl your tongue isn't because you've practiced for hours in front of the mirror. It's a genetic trait. And it's controlled by a single gene, so there's a tongue curling gene. Some people can curl your tongue, and if you can, then you have the tongue curling allele, that version of the gene that codes for a protein that allows the muscles in your tongue to curl that way. If you cannot curl your tongue, then you have the non-curling allele, a different version of that gene with slightly different genetic information that results in no curling. And these genes are certainly located on our chromosomes, on our large chunks of DNA. So each of our chromosomes is going to have thousands of different genes located along the long strand of DNA. So if we look at this idea of chromosomes and genes, remember for each chromosome we have a homologous pair. One set that came from mom's side of the family, one set that came from dad's side of the family. And so this is certainly going to apply to genes. We're going to end up with 2 copies of every gene. And these genes may be the same or they may be different depending on which alleles or genetic information you inherited from each parent. And so on this particular diagram, each of the little bands here represents a gene located somewhere on the chromosome. And so in this case, they show us 3 genes. Again, we'd have thousands of genes on a real chromosome. But in our little cartoon here, we've got 3 genes, and for each gene, we've got 2 alleles. And those 2 alleles may be similar or they may be different. There's a couple of important pairs of terms that are important to be familiar with too that again we're going to use an awful lot as we work through our genetics material. The first idea, what we were just looking at on that last slide, is referred to as the genotype. And a person or an individual's genotype refers to the actual genes an organism has. And so, for example, if I was thinking about your tongue curling genotype, it would be which alleles or which versions of the gene do you actually contain, which genetic chunks of information do you have. Now obviously I cannot see your genotype. What I can see is referred to as your phenotype. Your phenotype is your outward appearance or the characteristics an organism has. Usually it's something physical that we can see, but it could be something behavioral as well, like tongue curling, for example. Now obviously we're not just a collection of our genes, and our phenotype is made up of, or determined not only by our genes, but also by the environment. Now the environment is certainly really complex and really hard to measure on living organisms, and so typically as we go through our genetics chapter here in Chapter 9, we're going to pick traits where your genotype plays a much larger role in your phenotype than the environment. For example, our tongue curling. Obviously, it's mostly genotype, but the environment could play a factor. For example, if you got your tongue lopped off in some sort of accident down at the factory, it doesn't matter what your genes are, you're not going to be able to curl your tongue. Okay? But by and large, that's kind of an oddity, and so most of our traits we're going to look at, genotype is going to be the important factor in determining your phenotype. Now, if we look at your genotype in particular, you've got 2 options. You could be homozygous, which means you have 2 copies of the same allele. Again, homo as a prefix always means the same. And so if you're homozygous, that means you inherited the same version of that gene from both parents. Versus the other option would be heterozygous, hetero being different. If you're heterozygous, you have 2 different alleles for a gene, which means mom and dad passed on different versions of the alleles or different chunks of information that make up your genotype. And so your genotype can be homozygous or heterozygous. That genotype then combined with the environment will determine your phenotype. And any discussion of genetics and introduction of the genetics topics always starts with Gregor Mendel. And Gregor Mendel is one of the really important figures in biology and biological history. He's sometimes referred to as the founder or the father of modern genetics. Here's my obligatory Gregor Mendel picture. He was a monk. He did most of his work in publishing in about the 1860s or so, 1850s. And in the monastery, he worked as a gardener. And so part of his job was to tend to the garden. And one of the things that really got him interested in this idea of genetics was noticing that as he grew vegetables from one season to the next, certain traits seemed to get passed on, other traits didn't get passed on. Or perhaps a certain trait would disappear for a couple generations only to reappear 2 or 3 summers later. And so that really got him interested in this genetics topic. And in particular, his study organism was garden peas. And so kind of common peas that he was growing in his garden, he started to manipulate and to utilize for his experiments. And here we're going to see a common flower of a garden pea, and they make an excellent experimental organism because they're easy to manipulate and easy to cross. So one of the things that Mendel wanted to do with peas as his study organism is he wanted to do some crosses and cross different peas and see what traits were being passed on. And again, peas make an ideal study organism for this sort of thing. And so Mendel could pick the parents, decide who was going to be the father by taking pollen from the stamen, putting it on the carpel, which is the female part of another flower, so that was going to be the maternal or the mom side of the family, and then seeing which traits were being passed on. And these control crosses are really similar to what's being done today. Just take a little paintbrush, dip that pollen, and move it from one flower to another. Then if you snip out the anthers to the maternal flower because you don't want it to pollinate itself, cover it with a little paper bag, and then later on when those peas develop, we know, again, which parent was the father, which plant was the mother. And so he just did this. He would grow up the peas and observe which traits were being passed on. And he did this for many, many generations worth of pea plants in his little monastery garden. And within the biological community, Mendel is famous not only for his information and regard for genetics, but also because of the scientific method. Mendel was really the first person to apply the scientific method, as we've discussed, in terms of coming up with hypotheses, analyzing our data with statistics, to biology. Up until the middle 1800s, biology was mostly a descriptive science, describing what organisms looked like, or where they lived, or dissecting them and describing their parts. Mendel was really the first to apply statistics in this scientific method, and in fact his experiments today are still a model for how to go about designing an experiment. He was a very meticulous record keeper. Now, because Mendel was starting from scratch, he did pick traits that were relatively simple. And so here's a chart showing some of those traits with his pea plants. For example, they were easy to observe, and they were traits that only had 2 choices. Either pea plants have purple or white flowers, yellow or green seeds, tall, viney plants, or short dwarf-sized plants. And so he took this information, did his controlled crosses, and in that little monastery garden did over 50,000 controlled crosses, keeping track of which traits were being passed on, using statistics, looking for any pattern. And based on all of those crosses in his analysis, Mendel came up with 3 basic ideas. Now, he didn't call them laws. For him, they were hypotheses. Today, 150 years later, we call them Mendel's laws of inheritance because we see that these ideas have held up and certainly hold the test of time. Now, today, as we talk about these laws, we're going to use our current terms. Remember, when Mendel was doing his work, he didn't know what DNA was, or what cells were, or chromosomes were, or alleles, or genes. All that is certainly much newer. And so we're going to apply these terms to Mendel's basic ideas. And his first law is the law of dominance. And what Mendel said was that for each trait that we have, there are 2 different alleles, or 2 different forms of the gene. One of those is referred to as the dominant allele. The dominant allele, remember all organisms are going to have 2 alleles for each trait. If you have just one of those alleles is the dominant, that allele is going to be expressed. And when we say a gene or an allele is expressed, what that means is that that gene or allele is going to show up in your phenotype, that we can visibly see it. So if you have one dominant allele, you will visualize that allele in your phenotype. It will be a visible sort of trait. And because that dominant allele will show up in your phenotype, the second or other allele that you have is going to be masked or covered up. It doesn't really matter what the other allele is. If one of them is dominant, it's going to cover up that other or second allele. And that allele that tends to get covered up is the recessive allele. The recessive allele, the only way we see it show up in your phenotype is you have to have both of your alleles being recessive. And if they're both recessive, then you'll have that particular phenotype. So if an individual has one dominant and one recessive allele, that dominant will mask or cover up the recessive, and we wouldn't even be aware that that recessive allele was there in a genotype because we're not going to physically see it. So let's walk through one of Mendel's examples. Now today, by convention, anytime we talk about a gene or a trait, we assign it a letter, and it's just an arbitrary letter that we're going to pick. But we're always going to write the dominant allele as a capital letter. And so whenever we see a capital letter in our genetics, we know that refers to a dominant allele. The recessive allele is always going to be written as a lowercase letter, as the same letter, but in the small size form. Now, we can't look at a trait and decide, well, that's dominant and that's recessive. The only way we can figure out which alleles are dominant or recessive is to do lots of controlled crosses like Mendel did and figure out which allele tends to cover up the other. And so let's take a look at one of Mendel's examples. The most obvious thing he looked at was flower color in pea plants. So we're going to pick a letter. We'll just pick the P gene because that's what the book picked. And there are two different alleles for the P gene. There's the purple allele, and that was the dominant allele in his crosses, and so we're going to call that capital P for purple. And there's also a different version of that gene, the white allele, that codes for having a white flower color. Now that turns out to be recessive, and so we're going to call that little p or lowercase p. And so here's our two different alleles for our gene, the dominant purple, the recessive white. So if we apply that to the pea plants, here's our different phenotypes. Either pea plants have purple or they have white flowers. So let's compare genotype to phenotype. There's only three options for genotype here. Some plants might get two copies of the purple allele, so they would be homozygous for purple. Some plants might get one of each, have a purple and a white allele. Now it doesn't really matter how we write it, purple and then white or white and then purple, it's the same thing, having one copy of each. Although by convention we usually write it with the capital letter first, just because it looks nice to have a capital letter at the beginning of that. But again, genetically it doesn't really matter. And then finally, we may have a plant that is homozygous or has two copies of the white allele. Now obviously, if a plant has two purples, it's going to be purple. But where Mendel differed is, what about this heterozygous? Most people working at the time of Mendel said, if we mix purple and white together, we should get a light purple. We should get a different phenotype, kind of a blended phenotype. But Mendel said, nope, based on my experiments, purple is dominant, so purple is always going to cover up white. And so it doesn't matter what that second allele is. And so if we look at our pea plants up there, that purple plant, it could be big P, or big P, little p. We're not sure just by looking at it. And then the only way to get that white plant would be to have two copies of the white allele, little p, since it's recessive. And if we apply our terms, two of these are homozygous. The top one there is homozygous purple, or we could have homozygous white, and then the middle one there having one of each would be our case of heterozygous. Mendel's second law is the law of segregation. And this law refers to what happens when these pea plants with their two alleles are going to start to reproduce and produce gametes, eggs and sperm. And in particular, Mendel said that the two alleles that an organism has are going to segregate or separate during gamete formation, such that each gamete is going to get one allele. So if a pea plant had a purple and a white, it was heterozygous. Some of its gametes are going to get the purple information, some might get the white information. And then during fertilization, when egg and sperm fuse together, each gamete is going to carry its one allele along, and that's going to return us back to our two copies of each gene. And this should sound very, very familiar to us. We just talked about this last unit in Chapter 8, that it was a process of meiosis that takes our diploid cells having two sets of genetic information and generates our haploid gametes. And again, Mendel was describing this long before we knew what meiosis was. He didn't know how it happened, but he knew this must be happening to get the pattern that he saw. So let's take a look at this idea of segregation and how it's going to apply here as we start thinking about inheritance and passing on traits. So sticking with our flower color example, let's say we've got one parent who is heterozygous, so it's got a purple and a white allele for flower color, and the second parent has two copies of the purple, so it's homozygous for purple. Now during the cell cycle, we know that cells are going to copy their genetic information, and so each chromosome would have two identical copies of the sister chromatid, and then during meiosis, those cells are going to divide into 4 gametes. One cell divides into 2, and then 2 into 4, and that takes us from our diploid to our haploid. We go from 2 sets of, in this case, flower color information in our cell, so our gametes only have 1 copy of flower color information. And if the original parent up here, parent 1, is heterozygous, we would expect, on average, half of its gametes to contain purple flower color information, and half of its gametes to contain white flower color information. So half big P and half little p. On the parent 2 on the bottom, since it only has purple flower color information, obviously all of its gametes are going to get the purple information. They're all going to get big P. And then when we have fertilization occurring, one possible option here, our offspring between these two parents, would be a white containing gamete gets together with a purple containing gamete, so that an offspring might be heterozygous, having purple and white. And again, since purple is dominant, the offspring would be purple in color. The reason for segregation is meiosis, we certainly know today. And again, it's because these genes are located on chromosomes. And so if we've got, in our example here, a big P, when those chromosomes split in meiosis, one gamete is going to get one of those chromosomes, the other gamete is going to get the other chromosome. Certainly one of the powers of Mendel's laws is they're going to allow us to do genetic crosses and make predictions about what sort of offspring we might expect from one generation to another, whether it's pea plants or whether it's in humans trying to predict diseases. What are the odds of me having a child with a certain disease if I know, for example, that the disease runs in my family? And his first two laws, dominance and segregation, are enough to allow us to do some predictions and do genetic crosses if we're following just a single trait. And so let's walk through a couple of basic genetic crosses here. The parents we start with are always referred to as the parental, or sometimes the P generation for parental generation. So in this case, let's do basically a repetition of some of Mendel's crosses. He took what he called true breeding lines of plants, and true breeding meant homozygous. So he had a homozygous purple, two big P's, and he crossed it with a homozygous or a true breeding white, two little P's. Now in terms of phenotypes, one plant looked purple and one looked white. So the first thing we need to do with our crosses is always figure out the gametes. What genetic information is going to be passed on? In this case, it's relatively simple. That purple parent is always going to pass on purple or big P information in its gametes. The white parent always going to pass on white or little p. And so in this case, what we're going to see in the first generation, which is sometimes written as F1, so if you see that F1 notation in your textbook, or certainly you will see it in your lab book, that just refers to basically the children, that first generation of those parents. In this case, our F1 are all going to be the same. They're always going to get purple from one parent and white from the other. So they're all going to be heterozygous, and they're all going to look purple. And again, this was one of the confusing things about genetics that kind of got Mendel started, is how can I cross purple and white, and they're all purple. The white seems to have disappeared. And the way Mendel explained that was the white is still there, but it's being covered up. It's recessive. We're only going to visualize or see that dominant purple color. If we continue on, again, replicating some of Mendel's crosses, next he crossed two of the heterozygous offspring. And so he took two plants that both looked purple in terms of their phenotype, but had a purple and a white allele. Now, in terms of the gametes here, as we said before, if we've got a heterozygous individual like this, we're going to expect half their gametes to be purple and half to be white on average. Okay? Now, we can go through and try to pair up and say, well, if this purple sperm got together with that white egg, we'd expect this particular offspring, and kind of mix and match on our own. But the way we want to do this, that's going to allow us not only to make sure we don't miss any or skip any combinations, but that's going to allow us to have the right mathematical amounts of offspring we might expect to see, is by using what's called a Punnett square. And you may be familiar with Punnett squares if you've had a high school biology class recently. Now, Punnett squares aren't a magic bullet or a magic answer here, but Punnett squares are going to allow us to predict the offspring that we're going to expect to see, but they only work if we already know the gametes. Notice we've already figured out the gametes first. Now we're going to apply that to the Punnett square. And every Punnett square is different, and so if you learn to draw a little box and fill it in high school, we'll try to break you of that habit. The best way to draw a Punnett square is just to draw two lines. Draw one kind of horizontally and one near the edge, vertically. And then on the top, we're going to list one parent's gametes. In this case, a purple or a white. And we're going to separate those two things with a line because it's going to pass on one or the other in each gamete. And then on the side of the Punnett square here, we're going to list the other parent's gametes. Again, a purple or a white, and we'll separate those out. And now we can just mix and match. Sometimes the two purples might get together. We might have a purple and a white. Here's another purple and a white, another way to get that combination. Or we might have two white alleles. And so in this case, if we cross the purple and the white heterozygous with another purple-white heterozygous, what we're going to see is a 3-to-1 ratio. 3 purple offspring for every 1 white offspring we would expect to see. And so again, Mendel explained all this by doing these controlled crosses. Looking at a single trait in a cross is called a monohybrid cross, mono for one. And this is a replication of one of the traits and crosses that Mendel had done. And if we look at it all together here, again, one of the confusing things about genetics is we're crossing purple and white, and the white disappears. We get all purple. But now we cross two purples, and all of a sudden we get this 3-to-1 ratio, and we see some white offspring out of nowhere. And other biologists couldn't explain that. And not only did Mendel explain this concept first of dominance and recessiveness and segregation, but when Mendel published, people really didn't see his work. It was written in German. It didn't get translated very often. And by the time he died, people still hadn't discovered his work. And his work really wasn't discovered until about 1902, 1903, when people still had not figured out genetics. Some 40 years after Mendel published, it hadn't been figured out. And biologists came across his manuscript, and they actually reran his experiments and verified his results. And so while Mendel did this work in the 1860s, his work really didn't apply to genetics, and modern genetics really didn't start as a field until about 1900 or so, when his work was rediscovered. So let's take a look at this idea of these genetic crosses, and we're going to do a lot of these in lab throughout the semester. You always want to list the gametes, and that's the step everyone wants to skip, is to jump into the Punnett square right away. But always make sure you list the gametes parents are going to produce. Once you have that, then you can draw your Punnett square, and then you can go through the Punnett square and figure out, based on the genotypes, what phenotypes would I expect to see. So for example, take a minute and do this cross. So pause the presentation for a second and cross this heterozygous purple with a homozygous white and tell me what you get. The gametes that we would expect the first parent there to pass on half purple and half the time white. The second parent there only has white information, so all of its gametes are going to be the same. They're going to contain white flower information. So when we do our Punnett square, we're going to draw our two lines. We're going to list one parent's gametes across the top. In this case, we'll list that first parent first, either purple or white. The second parent we'll list on the side. In this case, it was just white. And we don't have to list it twice. If it only passes on white information, we just have to list it once. And then what we're going to see is half the offspring are going to be heterozygous, and half the offspring are going to be homozygous for white. And so in terms of phenotype, we would expect a 1-to-1 ratio, or for every one purple offspring they have, we would expect to see one white offspring as well. And obviously, while Mendel was working with pea plants, we know that his basic ideas apply to lots of other traits in many, many other organisms, and as humans, we're certainly no exception. And there's a couple I just want to mention as examples here, and I would call these kind of cocktail party traits. They're kind of interesting, they're easy to see, sort of like the tongue-curling example. They really don't mean much, though, in terms of health or phenotype. Something like freckles, for example, is what we would call a Mendelian trait, following Mendel's basic laws. It turns out there's a freckle gene, if we call that the F gene. Having freckles is dominant, so having freckles would be the big F allele versus no freckles, which would be little f. So if we take a look here, the girl on the left, she has freckles, so we know she's big F something. We don't know what her other allele is. She could be big F, or big F, little f. The girl on the right, though, we can see without freckles. Since she's a recessive, we know the only way she can have no freckles is to be little f, little f. Widow's peak is another common example here, having your hair come to a point in your forehead. We'll call that the W gene for widow's peak. Having the widow's peak is dominant, so that would be the big W allele, straight hairline, little w. So again, the recessive person there on the right, we know she's little w, little w. The woman on the left, she must be big W something. Okay? Now I do want to point something out here as we look at some of these traits. Notice that widow's peak is dominant and freckles is dominant. Well, that doesn't mean, though, that everyone's running around with freckles and widow's peak. And one common misconception is that if something's dominant, it's either better to have for you, or if it's dominant, that means it must be common. Everybody's going to have it. And that's absolutely not the case. That being dominant and being common are completely different things. They are not the same idea. Dominant and recessive only means if I have one of each, if I'm heterozygous, which one shows up in my phenotype. So, for example, having a widow's peak is dominant, but obviously it's not really common. In a group of 20-25 students, typically I only see 2 or 3 people with a widow's peak, which means there must be a lot more copies of that little w allele floating around than there are big W's. We've got one more law to look at from Mendel. His basic 3 laws. You have dominance, the law of segregation, and then finally the law of independent assortment. And this law of independent assortment really is not important unless we're looking at 2 or more traits at the same time. So if we're following several traits being passed on from one generation to another, we need to take into account this idea of the law of independent assortment. And what this law tells us is that the alleles of one trait, for example, widow's peak, are going to assort and be passed on independently of the alleles of all other traits. Okay? So the alleles of one trait assort independent of all other traits. And the take-home message there basically is, if I'm big W, little w, for widow's peak, if I pass on big W, that's not going to in any way influence what allele I may pass on at a second or third or fourth gene. That each gene is independent, and the alleles that I pass on or going to be simply random. If I'm big W, little w, I would expect half my gametes to get the big W and half to get the little w. And that's not going to impact in any way what I might pass on as a second gene, which alleles are going to end up in my gametes. Let's walk through an example then of this idea of independent assortment. And so we'll look at a dihybrid cross, which is just looking at two traits being crossed at the same time here. And so let's use a couple of Mendel's traits. Another couple of things he looked at, one was P shape, and we'll call that the R gene. Big R is for a round, nice kind of full round P, versus the little r allele, the recessive allele, is a wrinkled, shriveled up P. And so each P plant is going to have two copies of R. It could be homozygous round, homozygous wrinkled, or it could be heterozygous. Our second trait is P color. What color is that P going to be? And we'll call that the Y gene. The dominant P color is yellow, so we'll call that capital Y. The recessive is green. We'll call that little y. And so again, each P plant is going to have two copies of the Y gene. It could be homozygous yellow, homozygous green, or they could be heterozygous. And as we look at two traits, or even more than two traits throughout this unit, the basic genetics is the same. We'll have more letters to keep track of because we're keeping track of more genes, but the basic ideas don't change. And so let's look at a parental cross. Let's cross a parent that is dominant for all those alleles. It has two copies of the round information for P shape, two copies of yellow for color. And we'll cross that with a parent that's homozygous for all the recessives, two copies of wrinkled, two copies of the green. So our first thing to do is figure out which gametes are going to be passed on. And remember, each gamete gets one copy of each allele or one copy of each letter. So each gamete in this case has to get an R for shape and a Y for color. And so that dominant parent, since it only has round yellow information, every gamete is going to get a big R and a big Y. Likewise, for that recessive parent, every gamete is going to get wrinkled green information, or a little r and a little y. So this Punnett square actually is very simple. We'll draw our two lines. One parent's gametes on top. In this case, it only has one gamete, a big R, big Y, and the recessive parent only has one gamete as well. And so in this case, the entire next generation should be heterozygous. They should have round wrinkled information and yellow green, and because of the dominance issue, we would expect 100% of those offspring to be round and yellow in terms of their phenotype. Now, if we cross the heterozygous individuals, it is going to get a little bit more complicated, and the most common area where mistakes happen are the gametes. And so as we start looking at two traits or more than two traits, take your time and make a list of the gametes. And so, for example, if we were to cross big R, little r with big Y, little y, and cross it with itself, the first thing we would need to do is list the gametes. Okay? And so take a minute and think about what are some of the gametes that we might see produced here, remembering that each gamete has to get a big R and a big Y. And this is where this law of independent assortment comes in and tells us that one gene is not going to influence the other, which means basically any combination that's possible is a combination we would expect to see. And so, for example, sometimes we might get round and yellow information together in a gamete, the big R, big Y. Sometimes we might get round and green information, or wrinkled and yellow, or wrinkled and green, the little r, little y. And so we would expect all four of these gametes to be produced, and each parent we would expect 1/4 of their gametes to be each of these different types. And a way to look at this, if I was just looking at the R by itself and ignored the Y's, I would expect half the gametes to get big R and half to get little r. And if I look at my list, sure enough, half have round information and half have wrinkled. Likewise, if I cover up the R's and just look at the Y's, I would expect half big Y and half little y, and sure enough, that's what I see. And the reason we see this mixture of all these different gametes is independent assortment. So, for example, here's my cell that has big R, big Y, little y. Okay? As it undergoes meiosis, there's different ways these chromosomes may line up. If they line up with all the dominant alleles on the left, what I see is some gametes are going to get the big R, big Y together. Other gametes are going to end up after meiosis with little r and little y. But equally possible is we might get the case in alternative 2 where one of those pairs of chromosomes flips or kind of flops over and reverses. And if that's the case, that little subtle kind of flipping of the chromosomes results in different combinations of gametes. Here, big R with little y, or little r with big Y. And so this is our list of the 4 gametes we just made on the last page. Again, half of them are going to get big R and half little r, half big Y, and half little y. So if we look at our dihybrid example, with this heterozygous situation, we're going to have a lot more gametes, so our Punnett square is going to get bigger, but again, the basic genetics really aren't going to change. And so we're going to make our 2 lines for our Punnett square. list one parent's gametes across the top. In this case, we said there were 4 possible gametes, and we're going to separate them with a line because each gamete is going to have one of those 4 combinations. The other parent, since genetically in this case it's the same, we'll list those 4 gametes down the side, and again, we'll separate them with a line. Then we're just mixing and matching. Sometimes we might get those 2 dominant gametes together, and so we would get something that was homozygous for all the dominants, and so on. And so in this case, our Punnett square has 16 boxes, so we're going to get 16 different possible combinations based on our different gametes. Now, it gets kind of alphabet soupish. There's a lot of letters going on there. We don't want to really get sucked into that, though. What we really are interested in typically when we do these crosses are the phenotypes, right? If I cross this round yellow plant with another round yellow plant, what are their offspring going to look like, and how many might I expect of each type? So in this case, the obvious place to start is, well, how many offspring are going to be round and yellow and look just like the parents? And to be round yellow, they have to be big R something. Doesn't matter what the other R is. Big Y something. And so in our box here, if you go through and count, there are 9 different boxes out of 16. So 9/16, or a little over half, of the offspring we would expect to look like each parent, round and yellow. Now notice they have different genotypes, but they're going to have the same phenotype. Some of the offspring might be dominant in shape and recessive in color, so they might be round and green. In this case, we would expect to see 3 of those different boxes. Other offspring might be wrinkled and yellow, so they're recessive in shape but dominant in color. In this case, there's 3 of those as well, and then we've only got 1 box left over. That's for our all recessive, wrinkled and green. And so only 1 out of 16 offspring would we expect to see wrinkled and green in this case. And so by crossing this round yellow heterozygous with another round yellow heterozygous, we see 4 different options or 4 different phenotypes in their offspring. I just want to finish up going over a couple examples of some of these different crosses with 2 traits, because I know a lot of times they are easy to get tripped up on, and we'll work out these in lab as well. The first thing you want to do with any genetics problem, if it's a word problem, assign a letter for the gene. Sometimes you're given that information, but if you're not, just pick a letter, and it can be pretty arbitrary which letter you pick. Then make a list. What are the different alleles? What's the dominant? And I'm going to write that with a capital letter. What's the recessive condition? And I'm going to write that with a lowercase. Once you have that, you want to make a list of the parents' genotypes. And again, sometimes that may be given in the problem. Sometimes you may have to figure it out based on their phenotypes or based on other issues, what genes the parents have. But you always want to write that down. Here's what I'm starting with. Once you do that, then you're going to make your list of gametes. Here's what the parents can pass on. Then it's time for the Punnett square. Okay? Only after we've done those first 4 steps, and I know that intuition is to start with the Punnett square. Start with it first. The problem is if you start writing a square the minute you start your genetics problem, you'll probably stick something in the square, whether it's right or wrong, and that may get you into trouble. And so you don't want to do that Punnett square until you've already figured out what are the gametes, what are the parents passing on. Then you're going to do your Punnett square, and then you can go through the square like we just did with that last example and interpret the results, how many of each phenotype might I expect, and answer any questions that might be associated with the problem. So here's an example cross. Let's say we're going to cross a plant with purple flowers and round pea shape. So big P, big R, and let's cross it with a parent who has white flowers and round pea shape. Little p, big R, little r. So what I want you to do is figure out what are the different genotypes their offspring might have, which means do a Punnett square, and what is the ratio of phenotypes? What are the offspring going to look like, and how many of each might I have? So pause the presentation and go ahead and work that cross out. Once you've worked it out, what you should see is that each parent has 2 possible gametes it can produce. The first parent can produce purple and round information together or white and round information together. The second parent is going to pass on either white round or white wrinkled information. So when we fill in our Punnett square here, what we're going to see is half the offspring are going to be purple and round. The other half of the offspring are going to be white and round. And so we're going to expect a 1-to-1 ratio or 50-50, half of each type from this cross. Let's go ahead and add another letter. Let's look at all 3 traits that we looked at with Mendel: flower color, pea shape, and pea color. And so let's do this cross. Let's cross these 2 individuals together. Now don't let the extra letters bother you. That's just one more letter you're going to keep track of. Remember, each gamete has to get 1 copy of each letter. And so same thing for this problem. Pause the presentation, give me the information about what genotypes you would expect to see, what are the phenotypes you would expect to see from this particular cross. And the cross isn't nearly as bad as what it looks like there. The first parent has only 2 gametes, so we'll list those across the top. The other parent has 4 different gametes it's going to make. All the gametes are going to get little p, but then we're going to see the 4 different options we saw on our dihybrid cross with the R's and the Y's. And so in this case, there's going to be 8 options we might see with the offspring. 3 of those 8 are going to be purple, round, and yellow. So they're going to have 1 big P, 1 big R, and 1 big Y. 1 out of 8 are going to be purple, round, and green. So a big P, a big R, and 2 little Y's. 3 out of 8 are going to be purple, wrinkled, and yellow. So a big P, 2 little R's, and at least 1 big Y. And then finally, 1 out of 8, the last one there at the bottom right, is going to be purple, wrinkled, and green. Have a big P, and then be all recessive. And so using Mendel's laws, it's going to allow us to make these predictions on what we would expect to see, whether we're looking at 1, 2, 3, or even more traits at a time. So wrapping things up today then, Mendel developed his 3 laws of inheritance, again, way back in the 1860s or so. The first 2 laws, the law of dominance and segregation, if we use those laws, that allows us to make those single trait crosses or monohybrid crosses looking at one prediction. His third law, the law of independent assortment, applies to looking at multiple traits and allows us to do crosses looking at several traits all at the same time. Our Punnett square is our tool that's going to help us predict the offspring, although we're not going to use that tool until we've already figured out what gametes the parents are going to pass on. We know that there are many, many traits that follow Mendel's basic inheritance laws, whether it's plants like pea plants or whether it's humans or other animals as well. And make sure that you practice these crosses and you can do these monohybrid and dihybrid crosses. Again, we will practice them in lab. There's also some practice problems associated with your book and the website.